All right, welcome to day one lecture. Today, what I want to go over is just a little introduction to biology, what makes something living, and then I'm going to discuss the scientific method, how to collect data. And then your assignment today is going to be to answer just some basic questions about the lecture, making sure that you understood it. So what you do is you listen and you watch and you fill in the blanks of the actual papers that I gave you in your little class manual. And then you get on Teams and I have some questions. They're like general understanding questions to make sure you followed along and that you understood what I was talking about. So what you'll do is you'll electronically fill out that document and submit it. Then you have a lab that's called Is Yeast Alive? And so you'll electronically fill that out and submit it. So the only thing that you're actually writing down on paper are your notes, and then everything else is done electronically. I think you'll end up getting the hang of everything after a few days, but I'll just go over it again each day, and then I think after a couple of days you'll get the hang of it because every day is going to have the exact same structure. All right, so we're going to talk about the characteristics of life. So what is biology? What makes something living? So biology is the study of life. So we need to look at what exactly defines life. And so you've probably heard, especially with everything going on right now, is a virus living? You've probably heard someone ask that question before. And that's actually not a really simple answer. At least I don't think. As a biologist, I don't think that's a really easy answer to give. A lot of people will tell you, no, a virus is not living. You're crazy to think that they would be living. But it's my opinion that a virus actually kind of represents something that is like an in-between of something living and something non-living. It's like the transitional form. And so some scientists, some evolutionary scientists will ask the question, did we evolve from viruses? Did viruses evolve from us? And so it's just something really interesting to think about and talk about. But for those scientists that say, no, absolutely not, a virus is not living, they are going off of this traditional set of characteristics that define life. And this is your list right here. So all life contains order. And I'm going to go over each one of these in a little bit more detail, but I'm just going to give you a broad overview right now. So all living things contain order. And so if you think about yourself, you contain a certain order. You're made up of cells, which make up tissues, which make up organs, which make up organ systems, which make up an organism. All living things adapt, have evolutionary adaptations, respond to the environment, have internal regulation called homeostasis, have energy processing and metabolism, have some form of reproduction, and also growth and development. All right, so let's just kind of start from the beginning here and talk about each one of those. Living organisms all show order. So we're highly organized structures containing cells. And so you have some organisms that are just unicellular. They're made up of one cell. But even if something's made up of one cell, it still is going to show a certain kind of order. And as we talk about cells in the upcoming days, you're going to see what I mean by that when we discuss organelles, metabolism, and stuff like that. There's a certain order. So an example of the order that some unicellular organisms show, if you look at a paramecium is a single-celled organism. And if we are in the traditional classroom, we would definitely be looking at these under the microscope because they're kind of fun to watch swim around because they're so fast. But they don't have like a head or eyes or anything like that. So they'll just aimlessly swim all over the Petri dish. And then what I like to do is I like to get yeast and dye it with Congo red. And you can actually watch them eat the yeast and you can see the Congo red going through their stomachs. And it's cool as the pH changes as they digest it, the Congo red changes colors. So if you're taking AP Bio and we're in the classroom, we will definitely do that lab. But anyways, this is a paramecium, a unicellular protist, and you can see that there's little tiny hair-like structures called cilia, and that's what helps them swim around. So that's just an example of the order of a unicellular organism. If you kind of dive deeper into what the cilia look like, you can see that each cilia has a ring of what's called microtubules. So a ring of microtubules, and there's 1, 2, 3, 4, 5, 6, 7, 8, 9 around the perimeter, and then you have these 2 in the middle. And so that is what makes up the order of the cilia. And then if you look at the trachea like a human, you see the exact same order. The cilia of the trachea have the exact same arrangement of microtubules, which is really interesting. And this is definitely something that would be used to prove evolutionary relationships between humans and unicellular organisms like paramecium. So that's what evolutionary scientists would use as proof that we evolved from a common ancestor, because our cilia and their cilia represent or have the exact same order of microtubules, which is pretty interesting. So this says the internal structure of cilia, whether from paramecium or human trachea, have a nearly identical internal order. Super cool. And so this example right here isn't on your notes, I don't think, but it's just something interesting to mention. This is called a male Chilean stag beetle. It's also known as a Darwin beetle. And so what they have, as you can see here, are these crazy oversized jaws. And so these jaws are really crucial in its objective to secure a mate. And so a lot of times they'll climb trees really high searching for a female. And then when it finds a female and it sees other males in the vicinity, they will actually lock, that looks like horns. They'll kind of lock these horns right here and they'll fight, sometimes to the death. And so that just represents an organization and order, because you see structures like this actually in mammals too, that will fight each other, usually in hopes of securing a female mate. So what you will also see is that individuals with favorable traits will survive and reproduce. So the stronger these things are right here, the more likely they're going to be able to mate and reproduce. And these are their jaws, actually. They're not horns, they're their jaws. And so the stronger they are, the more likely they'll be able to fight and they'll be able to reproduce. So this could even be considered an adaptation. The bigger they are, the more likely they're going to fight and reproduce. All right. Another characteristic of life is responding to a stimulus. Living organisms have the ability to respond to changes in their environment called a stimuli. And I'm sure everyone has seen a Venus flytrap. In the greenhouse, I like to keep Venus flytraps. They're okay, at least for me, they're kind of difficult to keep alive. They require a lot of care, I find. I find them to be really high maintenance, and I haven't really been able to keep one alive. But when I do, or when a student, I've had students try to keep them in the greenhouse before, and it didn't turn out that well. But for a time being, we will get fruit flies, flightless fruit flies, those that have been bred to not have wings, and we'll put them right here, kind of like this fly. And when they sense movement, they actually collapse and then they'll digest the fly. So this is a Venus flytrap responding to a stimuli, another characteristic of life. Living organisms must maintain a specific range of internal conditions in spite of outside changes. And this balance is called homeostasis. I'm sure you've heard that word before. Chickens pant to give off excess heat. So they pant, kind of like a dog does, but I'm sure you didn't know chickens do that too. As humans, we don't pant. That would actually look really funny if we walked around doing that when it was really hot. But what we do is we sweat. So that's an example of homeostasis. We can also see homeostasis in humans in regards to blood sugar. When your blood sugar gets too low, you have this chemical or this hormone called glucagon that will access glucose in your liver, causing your blood glucose to go up. If your blood glucose goes up too high, then that's when the hormone insulin gets released, and insulin is going to either store glucose in your liver or it's going to make your cells take it up and use it to make ATP. So homeostasis is just that internal balance, and you see it with all living things. All life is able to reproduce, passing their genes along to healthy, fertile offspring. All right, and I think that goes without explaining. I mean, even a bacteria is able to split into two. One bacteria splits into two bacteria. It's undergoing a process called binary fission. And you know that mammals will reproduce, um, and fish will lay eggs, and all of that good stuff. I don't think it's really necessary for me to go over this, but if it's a living thing, it will reproduce. And that what's confusing is we'll see viruses reproduce. And so we have yet to get to something that a virus actually can't do. Organisms grow and develop along a specific sequence based on instructions present in their genes. And this is just showing you from an egg all the way through the tadpole stage into a young frog. So all living things are going to grow and develop, even a bacteria. Even a caterpillar changing into a butterfly. Oh, this reminds me, right before we left school. It's kind of a sad story, but that's okay. I like to put stories into my lectures to try to make them a little bit more interesting. Right before we left for spring break, we had caterpillars, and all the students each in a group. We had 14 butterfly houses, and they all had their caterpillars, and they were all in their little cocoons. And we went on spring break, and they never got to come back, but their butterflies did hatch, and I released them, and I had a little butterfly release Zoom party. I think they were kind of depressed by it, but at least it gave me a little bit of closure. Anyways, each organism must have a source of energy. So this is where a virus will not fit in. Viruses do not eat. They do not have a metabolism. They do not use ATP. And so I think this is the one thing that scientists will use as their rationale for a virus is not a living thing. And so those are all of the characteristics of life. Let's talk a little bit about the organization of life. So I want to talk with the broadest, most inclusive level, and that is the biosphere. So the biosphere is the biggest, broadest, most inclusive level of life. So it is the part of Earth that supports life. So it's like all of Earth and going upward to where birds and bacteria and anything cannot exist anymore in the sky. So all the way down in the earth, in the dirt, up to the furthest part of the sky where birds and bacteria and anything else can't live. Biosphere, because there's only one, is made up of ecosystems. Ecosystems are specific areas in the biosphere that have a common set of physical characteristics. So an example of an ecosystem would be the savanna, or the rainforest, or the deep ocean, or an estuary. So those are examples of ecosystems. An ecosystem is going to contain all the abiotic and biotic factors that contribute to that area. So all the living and non-living things that contribute to life existing in that area. So climate, soil, landforms, and all of the life. Ecosystems are made up of communities. Communities are made up of all the different populations of living things interacting in one ecosystem. So all of you can see right here, this is a community. So population of rhinoceros, lions, vultures, elephants, giraffes. So all of those animals living in one area. A population now, we're talking about all the members of one species that live near each other and are reproducing. And so this would be considered a population right here. So they're all the same species and they're reproducing. So populations make up communities. And then an individual organism is going to be part of a population. So if you can kind of see how there's like this hierarchy, okay? There's a hierarchy. Biosphere is made up of ecosystems. Ecosystems are made up of communities. Communities are made up of populations, and populations are made up of organisms. So that would be part of our study on ecology if you're going through a traditional biology class. We can take it a little bit further and say organisms are made up of organ systems and organs. Organs are made up of tissues. I'm going to give you a little bit of time to write this down. Tissues are made up of cells, and then cells are considered the smallest unit of an organism that still demonstrates all the basic characteristics of life. So that's important to remember. The smallest unit that can still demonstrate all of the basic characteristics of life. And this is just an example down here. This is moss. Moss is an organism that's made up of stems and leaves. So stems and leaves are actually organs of a plant. And each part, stems and leaves, each organ of the plant is made up of different tissues. And within the tissues, you have many different types of cells. Same thing with humans. And so how did biology begin? And you don't have this on your paper, but it's just worth mentioning that many of the original understandings of life came from philosophers who did not actually conduct experiments, but they used logic and reason. So they didn't have the tools that we have. They didn't have, they didn't have the microscope back then. They just didn't have all of the things that we kind of take for granted in a way. And so they really just had a blank slate. And so the first thing they used was logic and reason. And so this guy right here is Aristotle. And so he was an influential Greek philosopher. And he proposed that life could arise from non-living matter. So we're going to go through an experiment that was done to prove this was wrong. And so this is a quote from him. He said, "So with animals, some spring from parent animals according to their kind, whilst others grow spontaneously and not from kindred stock." Fancy, so fancy. And of these instances of spontaneous generation, that's what he called it, spontaneous generation, some come from putrefying earth or vegetable matter, as is the case with a number of insects, while others are spontaneously generated in the inside of animals out of the secretions of their several organs. And so what you should be thinking of, especially when he makes like these claims right here, is that when an animal dies, what is one of the first things you're going to see when an animal dies? And that's going to be maggots. And that is what he's referring to, is these animals are dying and then all of a sudden, out of nowhere, maggots are appearing. Therefore, there is a spontaneous generation of life from non-life because the animal's dead. All right. That is from Aristotle. I mean, good for him, for trying to find the answer. But he was wrong. And I'm going to tell you a little bit about an experiment that was done. And so today you're going to do a lab that's going to demonstrate the scientific method. And you all already know, because I'm sure you've taken a science class before, that yeast is alive. It's actually a fungus. A lot of people forget that it's a fungus because you think of a fungus as being like this moldy stuff that grows on bread, but yeast is actually a fungus used to make bread rise. You're going to use the scientific method and the steps to figure out, is yeast alive? All right. And so I want to talk a little bit about an experiment that was done that also demonstrates the scientific method. This was testing whether or not Aristotle's idea of spontaneous generation was actually true. So it says the idea of spontaneous generation was eventually discarded through the use of the scientific method. Thank goodness. Scientific methodology involved. So these are the steps of the scientific method that you will use doing your lab today. And then for those of you moving on to AP biology that are taking this class as the prereq, we will do a lot of experiments, hopefully in the classroom. Where we will work through all of these steps over and over and over. It'll be really cool. You get to design some of your own stuff, your own experiments. So the very first thing you do is observe and ask questions. You're going to observe the specimen. Okay, what am I working with? And then ask questions. From your questions, you're going to form a hypothesis or like a guess of what you think is going to happen. Then you will design an experiment. In a controlled experiment. And a controlled experiment needs to have a control, a basis of comparison. We'll talk about that in a little while. Then you're going to collect data. Then you're going to analyze data. So when you collect data, you're actually recording it in a chart. And then when you analyze data, so students get kind of confused in my class because data is just your table with your numbers. under analysis is actually where your graph goes, because the graph is the analysis part, because you see trends when you graph numbers. And then at the very end, you draw conclusions. And then I take it a step further, and I like for students to write out discussions. Now, you won't be doing any lab reports in this class, but just so you know, if you're moving on to AP, we do some lab reports, and I'll walk you through that pretty thoroughly. All right. So a scientific investigation begins with an observation. So we're kind of starting back over at the beginning of those steps, and I'm going to go through each one as it applies to the spontaneous generation experiment that was done. So the act of noticing and describing events or processes in a careful, orderly way is observation. A hypothesis is an explanation for an observation, maybe based on prior knowledge. So Aristotle's idea of spontaneous generation began with the observation that maggots would arise from meat or dead flesh. That's so gross. Fleas from dust, or tapeworms from other organisms. So he observed and he saw maggots seem to be appearing from nowhere. And so all scientific ideas, like I said, were based on thought and logic and not experimentation. So someone took it a step further, and they tested his hypothesis that life arose from non-life under controlled conditions. And they used independent variables and dependent variables. So it's really important to have an independent variable and a dependent variable and to know what those are. So the hypothesis should be tested using an independent variable. And then whatever change or effect, whatever variable is changed or affected, is called the dependent variable. So what I say is the independent variable is what you are manipulating. The dependent variable is what you are measuring. Any other variables should be held unchanged or constant. All right, so let's say we want to see, I'm just going to make something up, something that I do in my classroom. So let's say we want to hatch brine shrimp. Brine shrimp are also known as sea monkeys. So let's say we want to hatch sea monkeys, but we want to see at which salt concentration they hatch best. And so we're going to have a bunch of different petri dishes with a bunch of different salt concentrations to see at which salt concentration they're going to hatch best. So our independent variable would be the salt concentration, because I am directly controlling how much salt is going to go into each petri dish. So that's my independent variable. My dependent variable is, after 3 days, I'm going to count how many brine shrimp have actually hatched, and I'm going to record it in a table. So your independent variable is the variable that you are manipulating. Your dependent variable is the thing that you are measuring. And then any other variable that needs to be held unchanged or constant. So something that I would want to keep constant in my brine shrimp experiment would be, I don't want some of them to be warm while other ones are cold, because then that introduces a new variable that could have an effect on the outcome. So let's say I have 1% salt and they was in the sun, and they had more brine shrimp hatch than my 2% salt that was in the shade. Well, what was it that actually caused them to hatch? Was it the amount of salt, or was it the temperature and the amount of light? So you want to make sure that you keep all other variables constant so that you know exactly what caused your results. Okay, so then what you're going to do, and I need to move my frame down so you can see all the words. What you're going to do is collect data, and data can be quantitative or qualitative, and I'm sure you guys already know what those words mean. Quantitative is going to be a number, a numerical measurement, time, distance, number of brine shrimp hatched, number of maggots. Qualitative is going to be something that's a little bit, so if you are trying to prove or disprove a hypothesis based on qualitative data, it's going to be a lot harder to do that, because a lot of times qualitative data is based on perception and sometimes even opinion. So it's best anytime you can to figure out a way to record data that's quantitative, not qualitative. Although qualitative data, sometimes we just have to do it. There's a lab that we do in one of my classes where we measure the activity of liver, a liver enzyme, and we actually put hydrogen peroxide on a liver and we watch it bubble. I didn't have students set up an apparatus to measure how much oxygen, so they just ranked the bubbling from 1 through 5, not vigorous to most vigorous. And so that would be qualitative data, but that's less valuable than quantitative. Okay, so let's talk about this actual spontaneous generation experiment. And I'm sorry, my frame does not cover everything. So I'm just going to start at the top and then I'll move it down again. So an Italian physician named Francisco Redi proposed an experiment to test spontaneous generation. And so what he did was he made 3 flasks. All right, so he put a piece of meat. So remember, he is trying to see if dead flesh, if life can spontaneously rise from dead flesh. So he put this nice, delicious looking juicy steak right here. This was the control group. Because it was left open, there was no, there was nothing done to it. So it was your control group. Nothing's done to it. Kind of like if I was doing the brine shrimp experiment, I would have 1 petri dish with just plain water and no salt. All right. Then he sealed a flask. Then he covered a flask with gauze. And it's pretty obvious, you can see what happened. The flask that was unsealed, there were maggots that grew. The flask that was sealed, or the one that had gauze, no maggots actually grew, but the one with gauze, it looks like there was actually maggots on the top of the gauze. So what was happening with the meat is flies were flying in when nobody was looking, and they were laying eggs, and the eggs turn into maggots, and then the maggots turn into actual flies. All right, and so this just kind of goes through. So you have a control. Your control serves as a basis of comparison. And then the 2 additional flasks that were set up, 1 completely sealed and 1 covered with gauze to allow airflow. So if this is called your control, then these 2 flasks right here are called your experimental groups. So it's important to note that those are called experimental groups. So now that we have the word control groups and experimental groups down, how do the words independent variable and dependent variable apply to this experiment? So the independent variable is what the scientist is manipulating, and the scientist in this experiment is manipulating the covering of the flask. So the independent variable is the covering on the flask. The dependent variable is what is being measured, and what's being measured is the appearance of maggots. So the appearance of maggots would be your dependent variable. And as you can see, only the unsealed flask grew maggots. And his conclusion was, so he made his conclusion, that his hypothesis was correct and that Aristotle was wrong. Maggots did not spontaneously generate, but they grew from tiny eggs laid by these really sneaky flies that were flying in and laying them when nobody was looking. Conclusions are always going to indicate whether the original hypothesis was supported or rejected. In real research, you have to make your conclusions based on statistical analysis. And we do not do statistical analysis in cell biology, but we do it in AP. And for those of you that are not taking AP, that's fine. That's totally okay if you're not. But I do like to throw in there what to expect in AP for those that are taking it. So please excuse me if that doesn't apply to you. All right, so when I have students write a discussion on an experiment that they've done, I need them to include sources of error. So they did their experiment, they collected their data, they analyzed their data, they made a conclusion, and you cannot really form a conclusion without statistics, but in this class we are. And then at the very end, I like for students to tell me what kind of errors could have been done that could have influenced your results. What would you have done different next time? So did you use tap water had you used actual spring water or distilled water would have actually made a difference? Was there human error? Did you miscount something? Did you mismeasure something? Was there electronic error? Did you have some kind of machine that could have, glitched out or not recorded something correctly? And so I like for students just to make sure they know that no experiment is perfect. There's always going to be some source of error. And what that does is that sets us up to do the experiment better next time if we had the time to actually redo the experiment. All right, so basically a source of error is going to cause an error or otherwise cause an incorrect conclusion. So could your conclusion be wrong even though you saw a certain result? Yes. And it's always important to acknowledge that. And anytime you read like a legit study, like a peer-reviewed study, at the very end, you're always going to see them kind of circle back around and be like, yeah, well, this was my conclusion, but I could be totally wrong. So it's always funny to see that. You're like, oh, great, awesome. So it's never like a 100% decision on things, and science is constantly changing because of sources of errors and because of the redesign of certain experiments, right? So a probability is going to help predict the likeliness of an experiment result occurring simply due to random chance. And so this is where statistics come in. Okay, and so did I see a difference? Is it just maybe by chance that there are some maggots in the open flask and not in the closed flask? Well, sure. There's a chance that maggots just by chance, by some weird crazy chance, appeared in the open one and not in the closed one. We eliminate the idea of the flies laying eggs. And so what scientists use to help them kind of differentiate between was this based on chance or was it actually a significant finding is they use statistics. And what makes more legit statistics and more significant results is going to be your sample size. The larger the sample size, the more likely your conclusion is correct. And so here's an example of one that everyone can understand. There's a new drug that's come out that's going to cure coronavirus. Which study are you going to believe? The one that tested the drug on 5 people or the one that tested the drug on 5,000 people? And so everyone would be like, oh, well, duh. The more people you test it on, the more likely you're going to get a really clear answer. And so it's the same with every experiment. The larger your sample size, the more legit your conclusion is going to be. All right. And an example, and I guess it kind of relates. So Dr. Edward Jenner is famous for discovering a first vaccine for smallpox. Initially, his results were questioned by the Royal Society of Medicine because he had tested the vaccine on a single patient, which was his gardener's son. Lucky him. Actually, lucky him because I'm pretty sure it worked. The results could have been the result of a random chance. So this one person was given the vaccine and he ended up not getting smallpox. But perhaps the child was already immune. And so the results were only accepted after he found several other patients who volunteered their children, including his own, to increase the sample size. I mean, obviously in this situation, it turned out good. But, for a brand new vaccine that has been even rushed, I don't know if very many people are going to want their kids to get a brand new vaccine that's been rushed. And so that's kind of the dilemma we're facing right now with COVID is, a vaccine might come out, but are people actually going to volunteer themselves to get it? I do believe enough people will. I just talked to a physician's wife yesterday who told me that they are actually anticipating a vaccine to come out in October, and the first people that are going to get it are going to be the healthcare workers. So, I guess they're kind of our guinea pigs. No, in order to get FDA approval, they have to go ahead and treat and have people volunteer to get it. So people will have already been tested with it. All right, another major problem with experiments is going to be when scientists or patients influence the data to produce specific results. This is called bias. An example of bias would be, let's say, a drug manufacturer. Y'all, I talk a lot about drug manufacturings and medications because I got my degree in pharmacy, so that's why I always bring that up, just in case any of you don't know where my doctor came from. Not like a real doctor, I'm just like a pharmacy doctor. So let's say we have a pharmaceutical company that comes out with a new antidepressant, okay? And this antidepressant is gonna make them so much money, and so they're doing human trials on it, and they have a group of people, and they're giving the antidepressant to. And so the way that they find out whether a psychotropic drug works is usually by questionnaires. And so in a trial where antidepressants are being tested, the wording of the questionnaires can influence the way the person answers the question. And so that would be called bias. Did they word the questionnaire in a way that would influence the person to answer it in a certain way? So that would be called bias. Bias could also be presenting data in a certain way, distorting a graph. Let's say the graph, yes, has the correct numbers, but the scale is done in a certain way that makes it look like the results are really significant. That's done a lot, and especially in drug trials, that's done a lot. And so bias isn't always intentional, but it must be accounted for. And that's why we sometimes have blind experiments where the experimental subjects don't know whether they are in the control or experimental group. And then sometimes you'll have a double-blind experiment where the researcher doesn't know. So let's say this is the antidepressant trial. You have some people getting a sugar pill and some people actually getting the antidepressant. That's called a blind experiment. They don't know which one they're getting. So even if the wording on the questionnaires influence their results, because they don't know which one they're actually getting, in the end, if you're testing enough people, the real results will come out. A double-blind experiment is where the researcher doesn't know who's getting the placebo and who's actually getting the treatment. So that's pretty interesting. All right, so depending on the results of an experiment, a hypothesis may be rejected or it might be modified and new experiments are done and conducted. So you start off with your initial observation, make your hypothesis, you do your experiment, you collect your data, you interpret your data, you come up with your conclusion and you're just like, oh man, this was not that good. I need to make some changes or I need to change my hypothesis completely. And then you go all the way back to the beginning and you do it again. And that is science. So when we have a lot of people coming out right now saying, all these scientists are wrong, they keep going back on what they previously said, it's like, well, yeah, that's science. Especially when we're dealing with something that we don't know very much about. We have experience with it, and then we reform our hypothesis. And with, you know, and I don't want to get into like a debate with anyone about wearing masks or anything, but a lot of people will use statements made in the beginning of the pandemic, oh, masks don't work, against the people who are saying now, wait a second, we were wrong, masks actually work. And you really can't use that against scientists because it's a learning process. They don't know the answers from the very beginning. They reform and modify their statements and their answers based on experimentation and on experience. And so that's why, especially during this pandemic, it's been so frustrating with people changing their minds, is because they are actually going through this process and then looping back around and going through it again and then looping back around and going through it again. So that's why you hear some people changing their minds. I just wanted to bring that up. That's actual, real science. All right. Finally, at the very end, the experimental results are published, yay, in some sort of really, you know, awesome journal like Science or Nature or something like that, or some kind of medical journal, and it's peer-reviewed or reviewed and critiqued by other scientists. So how do you know that a study is legit? Well, it's going to be in a journal like this, or it's going to be in a database of journals. PubMed is like a really legit database for journals, or JAMA is a really legit database for journals. Science and Nature are too. But you know that a study is legit when it's been peer-reviewed. You'll have some studies that come out that haven't been peer-reviewed yet, and people will be like, wait a second, we can't take that at its word until it's peer-reviewed. So peer-reviewed studies that have been published in a legit journal are the ones that you can really take the results seriously. And if the results are accepted by the peers, then the hypothesis becomes a working part of the scientific community understanding. And then just lastly, and I think this is, I'm almost done, a hypothesis should not be confused with a theory. A theory is a large, broad explanation that's composed of many hypotheses and experimental results. So a theory would not be rejected by a single experiment, but a hypothesis could. So a theory is a large umbrella with a bunch of hypotheses underneath it. Okay, so an example of a theory is the germ theory, which states that some diseases are the result of the presence and action of microorganisms within the body. And so there's a lot of hypotheses and a lot of experiments that have been done to prove that one theory. All right, a scientific law is then a description of an observed phenomenon. So laws do not explain the phenomenon, they simply state it. They are based on repeated experimentation that have been, proven over time. So an example would be like Hooke's law of elasticity, Dalton's law of partial pressure, Newton's first law of motion, second law of motion, the law of conservation of mass. That's one that we'll actually talk about in the upcoming days. All right. And then lastly, you're done with your notes. I just wanted to talk a little bit about the different fields of biology. So we are going to focus on cell biology in this class. You also have genetics. We're going to talk a little bit about genetics, taxonomy, evolutionary biology, and lots of other different types of biology. Informatics, where people will analyze DNA, and that kind of leads into evolution. But lots of different types of biology and lots of different studies of biology, but it all starts with cell biology. And with that, I'm going to go ahead and end our notes. And so you guys are doing a yeast lab, and I will have explained the yeast lab in the Zoom, but I think it's actually pretty well explained on the document. So if you're doing this lecture early and we haven't had our Zoom, feel free to go ahead and start the yeast lab or wait until I give instructions in the Zoom meeting. I will talk to you guys later.